So we developed this procedure about 4 years ago in the monkey model. So basically we showed for the very first time that we could take a somatic cell, basically any cell from a body, in this case in the monkey, and we showed that this cell can be basically rejuvenated, or we call it reprogrammed, and become like early embryo-like cell. So this way we could produce this early, more potent stem cells that then now can be used to make someone's their own stem cells, and then use it, for example, for treatment of many diseases that are caused by for example, Parkinson model, where we lost some neurons, or diabetes, then we need, for example, replace cells that produce insulin. So in reality, we don't have these stem cells in our body that would make these tissues. But now we have a technique we would allow take any cell, could be skin cell, and make these kind of personalized stem cells for each of these patients. The purpose of patenting is because, you know, we as OHSU and then in my laboratory, we invested probably a decade of time to developing this technique. And of course, if it becomes clinical practice, so we hope that our technique will bring some dividends in terms of that money to research, so we could further expand this line of research. Another thing is, of course, so now we have to move these findings that we've done in animal model to human, which will require substantial financing, you know, using clinical trials, and hopefully this patent or maybe licensing of this technology to pharmaceutical companies, or maybe to some other you know, companies that will bring some money that we could invest into clinical trials and move this quicker and faster to clinics.